Dear children, today I am going to continue the science lesson. In the last lesson we saw the animal cell. We learnt about the parts of the animal cell also, isn't it? Now, in multicellular organisms, plants are also there. Do the plants have cells? And what all types of cells are there in the plants? We are going to see that in detail. So today, to observe the plant cell, there are different types of plant cells. So last year you have observed the stomata through the microscope, isn't it? Last year's 5th standard teacher would have shown you the stomata. So this time we are going to observe the plant cell. To observe the plant cell, if we use an onion cell, we can see it very clearly. So today we are going to observe an onion cell through the microscope. You all know an onion. What is an onion? We usually use this in the kitchen, isn't it? So today we are going to observe the onion cell. For that we need an onion. Then what are the things required? We need a microscope. Then we need, that day we used methylene blue solution. Today we are using safranal solution, that is a stain. We know it gives color to the cell. Then we need a slide and cover glass and an onion. So we are going to observe the onion cell. For that we have to cut the onion into two like this. After that we have to take the thin membrane of an onion. For that, when we remove it like that, it will come out like this, isn't it? So from here I'm just going to break it, and then when you remove like that, can you see? You will get a thin membrane like this. So that membrane, the cell can be observed on this membrane. You need only a very small part. So this I am going to keep it here. So before keeping it on a slide, what we have to do is, we have to prepare the stain. For that, I am going to put little water in a dish here. In that, we have to mix little stain. Just a drop of stain. So the stain is ready here. And in another dish, I am going to put plain water. We have to wash it also. So here we have this part, isn't it? Now this I am going to cut it. And I am putting it in the stain. It has to be there for some time, and the thin filament has to get the color. Now already the color has come. Now teacher is going to take this one, and I am going to wash it in water. Now we have to transfer it onto a slide. After that, what are we to do? We have to cover it with a cover glass. Now our slide is ready. I am going to keep it under the microscope for us to observe. Dear children, now you can see the onion cell under the microscope. I am focusing on that. You can see a wall-like thing, isn't it? Clearly, many things together. Each one is a single cell. And you can also see like a border line. Please observe it properly. Can you see it? Now we observe the onion cell under the microscope, isn't it? So here you saw the onion cell, you saw a border for it. So that is the cell wall which you can see, and inside you can see small dots. That is the nucleus of the cell. Now when we observe the animal cell, didn't we see that there are different types of cells in our body, isn't it? In an animal cell we learned there are epithelial cells, nerve cell, muscle cell and blood cells also. So animal is made up of different types of cells. So in the same way, are the plants also made up of different types of cells? You have observed it already in the 5th standard. We know that photosynthesis takes place in plants, isn't it? Photosynthesis takes place in plants. And through what is needed for photosynthesis? For the transmission of air, different types of gases. That is, the stomata which is present under the leaf helps in the transmission of carbon dioxide and oxygen. We know there are two types of transmission which happens in plants. One is photosynthesis. So during photosynthesis, what happens? Carbon dioxide comes inside the plant and it uses it for the process of preparing food and it releases oxygen. So for the carbon dioxide to come inside the leaf, what happens? It comes in through the small pores inside the leaves called as stomata, and the oxygen goes out through the same pores. And another transmission also happens in leaves. What is it? That is respiration, like how we all respire, the plants also respire, isn't it? For respiration, what happens? Oxygen comes inside the leaf and carbon dioxide goes out. That is same for all living organisms. So in plants there are two types of transmission. Now this happens through small pores inside the leaves. Now you have observed the stomata, isn't it? Now the stomata, they are small pores, we already know. That is protected by a cell called as guard cells. So the stomata is a pore, it has to close and it has to open. That is, during transmission the stomata has to close and open for the air to go in and come out, for the gases to go in and come out. Now that stomata is protected by guard cell. That guard cell we are going to observe under the microscope today. So to observe the guard cell we need a leaf. So here we can see leaf easily using a beetle leaf, because teachers not having a beetle leaf at home, I have taken the leaf of a yam. Clear stomata and the guard cells will be really clear. Here the under part of the leaf we have to take. So I have taken a very soft yam leaf from my vegetable garden. So this is how we prepare a specimen for observing the stomata. For that we have to take the leaf like this, and just what we have to do is we have to just tear it so that the thin membrane type layer comes out, like how we have taken the onion cell. In the same way we have to get a thin membrane. So for that we have to just hold the leaf like this and just tear it. When we tear it like this, we get a small membrane thin layer. Can you see? This thin layer is used to observe the stomata. Now this thin layer, you can just cut it, and again what are we to do? We have to put it in the stain. Now I have put that in the stain, and then I am removing it from the stain and washing it in the water here. After that, what has to be done? This has to be transferred onto a slide. So now the slide to observe the guard cell is ready. So now we are going to keep it under the microscope and observe. Children, here when we are zooming, you can see the stomata, isn't it? The center, can you see the cell there? That is the guard cell. In the center there is a black line. That is the stomata. The stomata is open now. And two sides you can see a bean shaped thing. That is the guard cell. And you can see the other cells also around it, isn't it? Like how we saw on the onion cell. So there are two types of cell which you can see through the microscope now. There are two guard cells and that is open. So the center, that hole which you can see, the small space which you can see is the stomata. So children, today you have seen the experiment of how to observe the plant cell, isn't it? So two types of plant cell you have observed. That is, one is the onion cell and the other one is the guard cell. So today's activity is you have to draw the onion cell how you saw in the thing, that is pictures given in the textbook, and also the guard cell, and write the experiment on how to prepare the slide and how to observe the plant cell under a microscope. So that is a work for today. Thank you.